And greetings, everyone. Welcome to the Evidence Reasons Academy. I'm Salvador Cordova, and I'm joined here by Katie. People know who I am, and everyone knows Katie just from her appearance on this channel. So this is our last recorded meeting before she goes back to school. And she has had an outstanding summer working as an intern and an assistant on some of the stuff on nuclear localization signals. This is an ongoing project. I think Andy McIntosh had requested that we not release a video that the three of us made, me, Katie, and Dr. Andy, because something slipped out of his mouth that could get him in trouble as he talks about stuff that's happening in the United Kingdom. And so he asked that even though we recorded a video, that we not publish it. And so I honored that. He's a colleague, and he's also a professor at the university where I'm trying to get a degree. And we are working, Dr. McIntosh and I are working on a paper on thermodynamics and statistical mechanics and the origin of life. So I don't know how I got down that rabbit trail, but so yes, he thought that our project is really, really, really important and would be a deal breaker for Darwinism. I have foreseen this. It is a field, like a lot of things I put on the table. Sometimes it's like no ID proponent or creationist has used this before, which is actually good. I like putting on the table arguments that people haven't used before, and that I think will be very effective and groundbreaking. And I've had the privilege of working with Katie to develop, to work on some research and to actually do research on nuclear localization signals. And I will talk about that in separate videos over time. But I'd like to congratulate Katie on outstanding work, and it certainly impressed a very, very senior faculty member, Dr. Andy. So, Katie, were you glad to meet Dr. Andy? Wasn't that fun?
Yeah, that was pretty cool.
Okay, so we're gonna, since we've completed all our projects, I promised Katie that this would be her day to ask me questions. With one provision, I wanted her to become acquainted with Clustal Omega and multiple sequence alignments. So Katie, if you could share your screen, backstage we had her set up to show Clustal Omega. And we'll look at it here.
Do you see it?
Yep, it takes about 5 seconds sometimes for it to appear. It may appear on your system before it does on mine. So well done. So have you done anything with multiple sequence alignments aside from BLAST in any of your classes?
No, not yet.
Okay, so you saw a little bit of BLAST where you were comparing a sequence in humans to see if it existed in Aspergillus or vice versa. And then also with P53 and zebrafish. And so there's comparisons. Now, there are times when sequences are very similar between species. So why don't you, I like protein sequences better than DNA or RNA because proteins can use almost the entire English alphabet. DNA, RNA just have those 4 letters, which is kind of boring to me. Okay, so it says choose file or use example. Do you see that? So
yeah.
Okay, just use an example here.
Okay.
So this is in the FASTA format, meaning they represent the amino acids with English alphabetic characters. Do you see that greater than sign? And then it says test 1 at the very top.
Yeah.
Okay, so that is treated as a comment. that's telling the computer, hey, this is just a comment. It's not part of the protein sequence, obviously. And then what you can do is you could take multiple sequences, like typically we'll take it from multiple species. So I don't know what these species are. They just grabbed it. And so we'll use default parameters here. So let's just, if you scroll down a little bit, just go ahead, give it a title. It's like Katie's first alignment. For the viewers out there, this is a free of charge. The government in Europe and also in the United States, for the benefit of society, they make a lot of biological tools available, which is great, for free. And this one's easy to use. So go ahead and submit. And there's probably ways you could save the work and the sequence alignments. But this is just to get you acquainted. Mm-hmm. Now, I'm actually surprised it took that long. That should have been instantaneous, but I guess it just hung up. So if you just scroll down, I like alignment with colors. Why don't you do show alignment with colors?
Oh.
Okay, so when it has dashes, that means, okay, obviously test 3 has 60 character, has way more characters. I'm actually not, let's see if that's correct. Test 3, I don't quite like the numbering. Mm-hmm. Okay. Oh, this is actually good. They have this, okay, they're actually the same, each of the sequences have the same length, but then it's going to try to find the best way to get things to align. Mm-hmm. And they'll put dashes where stuff doesn't, it doesn't seem to align at all. Oh. Okay, so this is kind of, okay, there's some smart guys that figure out, okay, what's the optimal way to get this, to kind of line up as best as possible, even if they're not identical. Mm-hmm. So that's something you would worry about when you start to get to really advanced levels. But this at least shows that these sequences are what we call homologous. Mm-hmm. Have you heard that word in school?
Yes.
Okay, so there is the evolutionary definition, which I think is flawed, but you have to learn it. And then there is like the original definition by Owen, Richard Owen, who actually coined the word. He said, things are built according to a certain plan. Mm-hmm. He actually appealed to a creator. So like if you see cars, like a Toyota car, like say a Toyota Prius is homologous to a Honda Civic because they're both cars. They're not exactly the same, but they have, they follow what you would expect of a car's architecture. Mm-hmm. So that was the original definition. Evolutionary biologists took Owen's original definition and made it refer to common ancestry. Mm-hmm. The creationists have said, look, it's not common descent, it's common design. So we don't say that a Toyota Prius physically, was given birth. It was a Honda Civic that gave birth to it. They're mm-hmm. independent, separate, manufactured objects. And so the ancestors of all the major groups of creatures were independent creations, but they have similarity. So, but since you're in biology, it's unfortunately been tainted and poisoned with evolutionary thinking when it's really not necessary. Mm-hmm. You could just say these sequences are similar. Yeah. We don't have to say whether it's common design or common descent. They're just similar. Sometimes it is common descent, like when we're trying to do paternity tests. So, do you have any questions? I mean, this is not that hard.
So I guess we could have also done this with our data, but it probably wouldn't have shown anything. Or would it have like tried to align it with
Okay.
any kind of similar?
Okay, this is a very good question. If you took a topoisomerase from a human and a chimpanzee, do you want to try doing it? You want to try to do a live test?
We could try.
Okay, all right. Let's see if we can do it in 10 minutes.
Okay.
Otherwise, we'll quit. So this is also still a good exercise. Let me take down your screen a little bit so you can get set up here. Hang on. Let me see. All right. So I'll let you. Can you get another browser up and then do top 2A, or how about P53?
Okay. Where should I get this from? Like Uniprot?
Uniprot? Uniprot. I'm sorry, you pronounced it correctly. Okay, is it ready? Okay.
No, not yet.
Sorry. Well,
Oh
you're on the Uniprot website, so let's.
yeah, I'm on the Uniprot website.
Okay, so we can expand it here. So
I see. Oh yeah, I see a chimpanzee. Okay, I see a human.
Okay, so the human is only 261 amino acids. The pan-troglodyte tumor antigen is 393. Okay, well, this looks like this could be fun. And then you have a mouse.
Mm-hmm.
So do you remember how to get a sequence? And you would cut it and paste it into a, like say, a text file?
Yes, I think so. You could just copy the sequence, right?
Yeah, you can, yeah.
Or download it.
Right, you can download it.
Okay.
And it might actually be better just to make 3 separate browser windows, if you know what I mean. One for the human, one for the mouse, and one for the chimp.
Okay, let me go back then.
Let me know if I need to take down your screen.
No, it's okay. I can just open in a new tab. Okay. So, should I hit resubmission for the...
Oh, yeah. Well, I learned something. I actually used a different. Okay, see, that's okay. Why don't you clear sequence? Okay. And you clear this. And now you can insert the whichever one you grabbed. You might want to say, do you know what species that is?
That was the chimp.
Okay. You could say chimp. You are so fast at this. This is great. This is heartwarming to see how well you're like a pro here.
Oh, I think I opened the human instead of the sequence. Okay, hold on. And then this one would be the human.
And this might be just the zinc finger portion, which is still okay. Okay, there is sometimes in Uniprot it's not clear, whether you're getting the whole sequence or a fragment, and that's okay. This is just for our, just learning Clustal, and it's actually good we don't have something that's just like 100% the same size. Okay.
Is that the same?
Okay, human.
I'm worried that I might have just copied and pasted. I think these are all the same sequences, actually. I think I may have made a mistake in the copying and pasting. Let me...
one way to find out?
Hitting...
Yeah, go ahead and submit. If they're all identical, but yeah,
okay.
It probably didn't.
I think they are the same, though. I'm looking at them right now. Or...
Okay, the mouse is different from the human.
Yeah.
That ends up with a D. Hmm,
Let me
PV.
just try.
Okay.
We'll see. Chimp, human, mouse.
Okay, so this is a lot bigger sequence. Let's see if it. I take a long time. A lot of the time is you're just put in a queue.
Mm-hmm.
You're waiting in line for other people's jobs to finish. Because, okay, so let's see what it looks like. Uh, let's do alignment with colors and show.
I went to. There it is.
Okay. All right, so when you have the stars, I think that means they're identical. And then the places, well, the coding will tell you.
Why does it? Or are these the lines that are similar? Because it's like, wow.
Okay, see, they're identical in position 1, the M, which is behind me. By the way, almost all, unless there's what they call post-translational editing, uh, the polypeptide will almost always start with M. That's the start codon.
Yeah.
But not always. Biology is full of exceptions. So do you see like where there's one column that doesn't have a star? You have AAV, so alanine, alanine, alanine chimp, and the mouse. And this does suggest, if you see where there's a difference, usually it's between the sim. Um, the human and chimp are almost identical.
Mm-hmm.
Actually, if you erase the mouse, you could see how. It'd be curious to see how similar we are to the chimp.
Yeah.
But well done. We did this in 7 minutes. So
Yeah.
okay, now this is why I get on the case of creationists. They're saying they're trying to say we're not that similar to chimps. And I tell them, I said, I have looked at individual sequences where we're 100% similar.
Mm-hmm.
So I'm really glad you're getting to see this firsthand and you have a chance to discuss this with me. Um, when you do more of these sequences, um, you're going to see a lot of similarity, and that is something to deal with. Um, but before we get to that more, philosophical question, do you have any questions? This is almost everything you need. There is a way to make a phylogenetic tree. Have you ever seen a phylogenetic tree? It's like a family tree.
Yeah.
Okay, so scroll down and see if we can make a phylogenetic tree.
Is it? I don't see anything.
Go up. It's
Guide.
there. Phylogenetic. Try that. Uh, it's supposed to. There you go.
Wow.
And it gets and I think the 0, 0 means there's zero differences.
Yeah.
100%. We can also test this. Are you game for a quick test?
Sure.
Okay. Just go back up to your original sequence and
Where's
delete the mouse sequence. If you scroll up.
where?
Did it take away your?
I put it back in.
Oh, try resubmission. Try resubmission. Okay, and scroll down. See if it'll give you your. Okay, so just go back in and delete the mouse, and we'll rerun it.
Okay.
You know what? I was wondering what we were going to do for our last session. Now I'm really glad we chose to do this.
Yeah. I also had a thought because when we look at the nuclear localization signals, they almost are like never the same. They're always usually really different between species. And my thought was that you could use something like this even if you have a lot of the protein seems to be similar, the part where it differs, probably towards the C terminus, might indicate like a nuclear localization sequence.
So do you see any differences?
No, not in this one.
Okay, so this also means nuclear localization. Okay, I'm sorry, I interrupted you. Go ahead.
Well, I was just asking, is this our whole sequence that we put in, though?
We've done this. This is such a fast attempt. I would say probably.
Because I'm not sure if it's just fragments.
This could be the zinc finger portion.
Yeah.
But it could be that the nuclear localization signals for chimps and humans would be identical here. This is something we're going to have to deal with. So do you have any other questions? This is a good place to transition to one topic I wanted to cover with you.
Okay.
So, but if you had any questions before we move on.
No, I don't think so.
Now you know how to make a phylogenetic tree. And
Yeah.
you can make a huge family. I mean, with only 3, it's hard to make a phylogenetic tree, but you can build them.
Yeah.
Huge numbers. So this is such beautiful work, and we could have more fun doing this, but we'll have more fun doing something else. I'm going to show you. I'm going to share my screen now.
Okay.
And let's see. So this is actually going to relate to what we just did with the multiple sequence alignment. So is that pencil really bent or not?
No, isn't it? The light is like refracting or something.
Yes, it's an optical illusion.
Yeah.
So the reason I'm showing you this, and I'm just sharing a personal anecdote here. It troubled me being a creationist. We look so similar to chimpanzees, and you saw it. So I get a little upset sometimes when creationists saying we're 15% difference. It's like, well, okay, globally we are, but for individual genes, it's, if you're sending off a creationist in college, they're going to see what you just did. I mean, they're going to experience seeing that for themselves. 100% identical. It's like, well, yeah, we're 15% difference, but we have, there's segments where we're identical, and it should give us pause. I'm not saying it's wrong. So it's just like when you look at this pencil, it looks bent, but it's not. But it's still, it took thousands of years before you figured out what was going on. Was it really bent or not? Because the data tells you one thing. If you only look at portions of the data, but you have to look at the whole. You have to examine all the data, and you realize it's not bent. There are numerous experiments that can show it doesn't actually bend, but it's still a problem why it looks bent. Do you see why I showed this illustration here? The common design at times can look so compelling to suggest common descent. And so there are kind of ways around this. If life is young, and we've asked evolutionary biologists, and you've got them when they don't want to answer it. I said, look, if life is only 6, let's just say hypothetically, I'm not even going to insist, but hypothetically, if life is young and has only been around for 6,000, or say 10,000 years, wouldn't that indicate common design rather than common descent? They feel very uncomfortable with that question. They'll say, well, that's irrelevant because the Earth life is obviously millions of years. But I would just say hypothetical. They get uncomfortable with that. So what do you think about the Earth? If the Earth is young, would you agree with what I was saying, that there can't be a common ancestor. There's just not enough time to evolve them. They would have to have been created separately.
Yeah, for sure.
Okay, and this is why I get on creationists. They're using all this genetic arguments and dissimilarity. I said, look, we have 15% difference between chimps depending on what you look like, how you look at the data. The different, if you align all the chromosomes, you would get all of them, which you really can't do. But we have algorithms like things called last Z,
Mm-hmm.
where you take, we have 3.3 gigabases, and I think the chimp has more. If you just, obviously, if the chimp has 15% more DNA than we do, we're going to have a mismatch.
Mm-hmm.
So how can that be that we have 100% similarity in some parts and then globally it's 15% difference? To some extent, it's like if we take two random books and we compare the spelling of the individual words, we'll say, hey, look, practically all 99% of the words are identical. So we have 99% similarity between this book and that book. Do you see the problem?
Yeah.
Okay, good. And so there is an active debate on the internet. How different are we really from chimpanzees? And I'd say, look, you could use one figure of 15%, but we've just looked at the P53, that segment, which I think is just the zinc finger segment. I could be wrong. And it's 100% identical. Or my colleague Casey Luskin brought an interesting point. I'm just trying to acknowledge him here. He said, well, even if we have some similarity, they'll still insist there's common descent because they said we're like 50% similar to a banana. He said, so obviously, so how similar, or how different do we have to be before you say there's no common descent? It's like, well, like 100% different, which is probably not going to happen. So I just wanted to say we're very similar. So people will say, well, then is God trying to deceive us? And I'll say, well, look at this pencil. Is God trying to deceive us by making it look bent? That's just a rhetorical question. I mean, just something to consider.
Mm-hmm.
I'm like, well, God has given us enough data to help us resolve the issue that the pencil is not bent, it's straight, there's an optical illusion. Therefore, God is not lying. It's just the way electromagnetic waves are bent in water. We can't say, God's being deceptive. And likewise, if we get markers that the fossil record is young or that life is young, then God has given us enough data to decide whether this is common descent or common design. So that is my answer. Now, if we adopt an old Earth viewpoint, it's going to be much harder to decide between common descent or common design. I would say we don't, even though I'm a young Earth creationist, I would say if we adopt an old Earth view, it's going to make the argument a lot harder to decide.
Mm-hmm.
But I would still say, like what we did with nuclear localization signals, we would need miracles that are indistinguishable from special creation to be able to make something as different as between a plant and a rabbit.
Mm-hmm.
To have a common ancestor. And that's a discussion for another day, but there's no universal common ancestor for all major protein families. If you ever start to get deeper into this, you'll find out that all major protein families do not align. So if you take a topoisomerase, it's not going to align with a collagen. They have separate independent origins. And even an evolutionary biologist told me that. So he agreed with me. So that being said, I'm now at your disposal. If you have any questions, I probably will say I don't know if I don't know. But if you have anything on your mind, just feel free to ask right now.
I'm trying to think of something to ask. I probably should have thought about this beforehand.
Well, I should have told you it was going to be a free session. I didn't even know what we were going to talk about. So, do you ever encounter people that, um, I mean, if you have questions about the strength of the creationist argument, okay, so I'm going to level with you. I don't think it's that good yet.
Mm-hmm.
I've seen it. What it was 40 years ago was so bad compared to today, but there's still room for improvement. So we could start there with, uh, if you've had questions on the creationist model that you think you're concerned about, that, is this a good argument? Do we have data on it? I can answer that. If you've been confronted by other people talking about evolution, we could talk about that. And that being said, anything else is free game. Ask me anything. It doesn't even have to be science, but we'll just start with science.
Okay. Have you? So I guess I'm just trying to think of like different arguments. Going back to like the evolutionary field, I'm trying to think of different arguments that people would make for like an old Earth because you're a young Earther. Have you seen the different like not fossils, but like skulls that people have like dug up that indicate that they might be transition kind of like human forms? Like I think they're called, I looked at their binomial nomenclature and it's like Africansis or something like that.
Yeah.
Is that familiar?
Yes. Okay. So I'm so glad we did Clustal, because I mean, you saw it, we're 100% identical for that section of P53 in the protein.
Mm-hmm.
Why even dig up fossils and argue over it? We are so similar. So are there transitionals? I have said, look, I would say not in the evolutionary sense, but in the conceptual sense.
Mm-hmm.
So if you look at, and I'll tell you what I think, again, remember that picture of a bent pencil. Is God deceiving us by making the pencil bent? I'll say no, because if you look at all the data, you can say that this isn't bent. So we can also look at the question of common design versus common descent. If the universe, if life is young, only 10,000 years, then it's common design. So the question would be, why would God make us so similar to chimpanzees? There are two important reasons. And it answers the question, is there a transitional? I'll say probably yes, only in the conceptual sense. So what you'll find as you do more sequence alignments, if you take, you could see that the mouse is a little bit different from us. If we took a plant, if we took another creature, I don't know that plants have P53. That's a good question if P53 is only found in mammals or animals.
Mm-hmm.
But if you took all the P53s out there, you could probably arrange them in terms of similarity to humans. You'd have chimps right up there, and then mice. And I don't know, if a whale has P53, I would think it'd be even more different, if you know what I mean.
Yeah.
We could take a rabbit. So, okay, so if we take any random creature, like say a whale, and then a human, okay, let's say hypothetically the whale is the most different from us. That's actually, I mean, this is, now that you have the tools, you can actually explore this yourself. Find out which creature is the most different from us in P53.
Mm-hmm.
Do you think you could find a transitional between, let's just say it was the whale and a human? Could you find a transitional? Meaning, can you find some sequence that's kind of, in between. It's somewhat similar to a whale and somewhat similar to a human, but it's kind of in between.
I'm sure
You get.
could.
You're sure you could, right? That's a conceptual transitional. It doesn't necessarily mean it's physical.
Mm-hmm.
So that's why I don't even bother with all these fossil records, because you could do it. You could definitely see it in molecular sequences.
Mm-hmm.
You could find billion transitionals. It's like, why do we even need to argue?
Mm-hmm.
Now, the reason that there's still some argument is sometimes we're not able to derive the protein sequences. Definitely, I mean, it's very, by the way, in case you didn't know, it's very difficult to get a protein sequence directly from the proteins.
Mm-hmm.
We have to get it from either the DNA or the RNA, usually the DNA in an ancient fossil. The reason is we have machines that can sequence DNA, machines that can sequence RNA, but we have to have just massive amounts of experimental machinery to actually sequence a pre-existing protein. It's the biochemistry and physics of it. So I think that's why they sometimes just rely on the bone structures and stuff.
Mm-hmm.
I know that was a long-winded answer to your question. I would say there are transitionals. You can build one. You can build transitionals all you want, but I mean, just because a rabbit might be in between us and a whale doesn't mean that a whale evolved into a rabbit and then into a human.
Mm-hmm.
So that's one of the problems with transitionals. Is like, well, what path are you going to take?
So then, would the transitional? This is definitely just theoretical, just something I thought of in my mind. Like, would the transitional be made more like the human, like intentionally made in God's image, and have like a soul and like all that stuff, compared to like an animal? Like, how would you describe that?
Okay, that is a beautiful, deep theological question. I cannot answer only God knows about souls.
Yeah.
And to my reading of the Bible, I have not gotten anything that said the animals have souls.
Yeah.
But that being said, they have, in the Old Testament, they use them as substitutionary. They use them. They're so close to us that they're sacrificed on the altar, and Jesus is called the Lamb of God. So a lamb, it's like, well, does it feel pain? It's like, I'm pretty sure it does. And it is a picture of Jesus Christ. But that being said, as far as do they have souls, where do the souls end up? I think only God knows. I mean, we have had certainly theologians weigh in on the question, but I think their guess is as good as yours or mine. So, but definitely, you could see that the animals are physically transitional, because we're more similar to a lamb or a cow than we are to a tree. And so when they describe the sacrifice of the lamb, either for Passover or any of the ceremonial, it actually describes all the organs. And those organs are homologous to the organs we have in humans. So I know you asked about souls. I have no answer for that. Your guess is as good as mine.
Yeah.
So,
I was just curious, like, what you'd think. Like, would it be more if they're transitioning into a human, would we treat them as an animal? Do they have like the same? Because their skull capacity, like, is obviously, from what you would see in the fossils, a lot smaller. Like, they have much smaller brains than us. So, like, do they have the same intelligence as us?
There are times animals have certain level of intelligence to do certain tasks. Definitely the birds can navigate the North Pole. Like, they can navigate North to South Pole, some of them. there is a bird species that can navigate nonstop from Alaska to Australia,
Yeah.
230-hour flight, and it has all the instruments in its head. It has having been a pilot, it's like all the charts you have to have and read to navigate, especially if you're out in the middle of the ocean. You have there is some sophisticated brain work and also sensory equipment the bird has that we actually have to build very sophisticated instruments like GPS, and in prior eras, sextants and uh, you know stellar navigation, and then we went to inertial navigation. I mean, it was just it's amazing what the bird has. So, but I did want to I want to throw this out to you.
Mm-hmm.
The transitionals are very important for understanding human biology.
Mm-hmm.
And you're learning a little bit of it since you did work in the lab. We learned about a lot of human biology by studying fruit flies.
Mm-hmm.
Because isn't it nice you can sacrifice those little creatures to help understand aspects of human biology? So let me give you a little history. We were able to decipher the genetic code, and let me see if I can just grab it here in the meantime, because we studied bacteria, and isn't it? I mean, the human genome, the way its transcription and translational process works, is so much more difficult to uncoil than it is for bacteria. And so we have to be very grateful that we are similar enough to, I'm trying to get a copy of the genetic code here for our viewers, that we can experiment with bacteria to learn the genetic code. So let me see if I can bring up my screen here. So this is the genetic code. Actually, it's the RNA one, not the DNA.
Mm-hmm.
Most of them are RNA. But this won Nobel Prizes just to figure out this little table.
Yeah.
That one, and we also began to learn. You've studied chromatin?
A little bit.
Okay. It's so nice to talk to a biology student because you know a lot. You've been exposed to this.
Mm-hmm.
We've learned a lot about human chromatin by studying yeast chromatin. So
Mm-hmm.
you can, as you work up the ladder, what we call model organisms. So we think they model a little part of the human biology. So as you work up the tree, kind of the conceptual transitional tree, you work up on creatures that are more and more like humans. But so why would we want to study a fruit fly instead of a chimpanzee? Or why would we want to study bacteria or yeast instead of like a chimpanzee to understand human biology? That's just a rhetorical question. I'll give you, why don't you try to articulate why you think that might be the case?
Why should you study? Wait, say the question again.
Okay, why should we study yeast or bacteria to learn about human biology instead of just studying chimpanzees all the time or humans?
I mean, from my experience, it's a lot cheaper to use, like yeast and bacteria, but I guess I know a lot of work that I've done with fruit flies. We can take something from the yeast and inject it into like a fruit fly to get something that we want, like a certain gene that we want to express or something like that. So you can use them as tools to further your research, I guess.
Well, I think you hit the nail on the head, and I'm just going to refine it a little bit.
Yeah.
It is cheaper. It's cheaper because it takes a long time to breed a chimpanzee or even a mouse. A bacteria will replicate in a few minutes, maybe 20 minutes, or I mean, when you culture them, you can get a bazillion of them overnight. And because they have small genomes, they're easier to work with.
Mm-hmm.
So it's nice when you can decompose what you're studying into smaller pieces. And also, if it's cheaper and it's less trouble. I mean, trying to raise a chimpanzee to study it is hard, right?
Mm-hmm.
So there are little pieces spread out over biology. You can learn a lot of biology just studying yeast and bacteria. Um, and and we've actually learned about the human nervous system by studying nematodes.
Mm-hmm.
C. elegans. We actually have some similarity to certain plants more so than other animals in their introns. My professor actually did a paper on that. So they're okay. This is getting a little theological. God has spread out little pieces of the puzzle to understand ourselves throughout all biology.
Mm-hmm.
Some creatures are more accessible than others to work with. So, we probably could have figured out the genetic code by working with human cells, but it was way easier to do it with bacteria. And we probably could have learned about chromatin in humans by looking at human cells, but it's way easier to work with yeast.
Mm-hmm.
So we can be actually grateful for all these what I call conceptual transitionals. I will actually say that I think Creation Ministries International says don't use the argument that there are no transitionals.
Mm-hmm.
Um, on the other hand, some creationists will be saying, would be probably arguing and saying, Sal, don't use the word transitionals. That's evolutionary. It's like, well, okay, whatever you want to call it. All right. They're definitely creatures that are more similar to us than others. I mean, in that sense, they're transitional. If you don't like the word, just make up some other word. So I hope that was helpful. I knew it was a real stumbling block for me personally, because when I started looking at all the similarities, I said, well, maybe we did evolve.
Mm-hmm.
But then I realized, well, if the Earth is young and life is young, then it's common design. And even if there's some intelligent design proponents like Michael Behe. Have you heard that name?
Yes.
He believes in common descent but intelligent design. So he's sort of a creationist, but not quite.
Mm-hmm.
Um, Even if there were common descent, there would still need to be the action of God to make it possible. Definitely in the prokaryote to eukaryote transition. That's just, I don't know how you can get around the need for miracles. And one aspect of that is nuclear localization signals. So I think this is the part that I felt I really wanted to. I'm really glad we did Clustal today, because this is, there are people that once they see that similarity, it starts to give them pause, and it gave me pause when I looked at it. I mean, when you see these protein sequences, and it's like we're 100% identical to chimpanzees.
Mm-hmm.
But it's not every gene is going to be that way. Just
Yeah.
for the record. Okay. I don't know. Have I at least given you good answers that you can work with for some
Yeah.
of that stuff? Okay. Any more questions? We have, we've normally gone till 3:30. Otherwise, you certainly email me afterward. I'm going to take time here just to let the audience know. I'm going to be referring Katie to Emily Reeves, who's at the Discovery Institute. Emily Reeves is publicly known with her association with the Discovery Institute. She's a PhD biologist, biochemist. She's also published in in an engineering journal, a prestigious IEEE, the Institute of Electrical and Electronics Engineers. So she has kind of a little bit of engineering background, plus she's a biochemist, PhD biochemist and biologist. So I'm encouraging Katie to build out her network of people who can mentor her and also give her advice on all sorts of professional and academic and maybe even personal mentoring. Um, it's also a chance for me. I'm mentioning Emily Reeves just to show that our audience, there's some really bright minds out there who are very accomplished scientists who believe in intelligent design. So the Discovery Institute is not specifically a creationist organization. They have people like Michael Behe who accept common descent and the long ages of the Earth. There is Answers in Genesis, which is a creationist organization. They've had kind of a chilly relationship with the Discovery Institute, because the Discovery Institute, has people who believe in common descent. I don't agree. I'm much more comfortable with the Discovery Institute. I do have some reservations about Answers in Genesis, but every creationist organization has, their own specialty, and we don't all agree, and that's okay. But there is an individual there by the name of Marisa Tillery. She had matriculated through academia, got her PhD in biology. She had brilliant work, and she would also be a good mentor for and someone to put in your network. So I just wanted to give a plug for my two colleagues there. And so now that I've made that back to advertisement, back to our regularly scheduled program, are there any other curious questions you had about, even science questions, not even creation related?
I had a creation related question, but
Okay, go ahead, anything. This is your
so
time.
I heard this one time as a a biblical defense of evolution, and I wanted to hear how you would refute it, I guess. So in Genesis, I had to pull it up. It says in Genesis 1, and God said, "Let the earth bring forth living creatures according to their kinds." Um, I'm going to skip down actually. And God made the beasts of the earth according to their kinds, and the livestock according to their kinds. I think the emphasis is on like let the earth bring forth, and that about the beast, and I think he talks about like the birds, and then he says, let us make man in our image after our likeness. And so I guess is that an evidence or just like biblical proof of, would you say, like when the Bible says let the earth bring forth animals, but it says let us make man in our image, humans would not fall under that? Like, what do you think about that?
Okay, so this is an excellent question, and the way I have resolved this, first I'll just relate it as a personal level. There was a comparable issue about whether, are you familiar with the argument between the geocentrists and the heliocentrists? Meaning, there used to be a belief that all the stars and the sun orbited around the Earth. That's called geocentrism, versus heliocentrism, where the Earth and the planets in the solar system orbit the sun. So I'm going to try to get to your question, but I wanted to set this up. So they tried to resolve this theologically. And so in the time of John Calvin, he said, Copernicus, who advocated the heliocentric model, these guys are of the devil. Okay, and they went through all of the Bible and said, the Bible teaches clearly geocentrism. And I'm like, well, I totally get it that you might arrive at that conclusion, because it doesn't really, my reading of the Bible doesn't explicitly suggest that we live in a heliocentric solar system. So why did I bring that up? Sometimes God is mysterious. He doesn't make the truth abundantly clear. In fact, that's an ongoing theme. I'm writing a book on finding the hidden God, because there are times God will not make it really, really obvious until later what the truth is. Certainly prophecies are mysterious. In the Bible, they say these are dark sayings. So why did I throw out heliocentrism versus geocentrism? I said the data will eventually help you understand what the Bible is teaching.
Oh.
And so what we have done, okay, so let's look at this. If we had the old Earth model, in common descent, we'd still say, well, God had to intervene because that's what the data suggests. There need to be miracles along the way. Starting with the origin of life, we know that the first cell is just improbable to come from natural processes, and I'm working on papers that actually describe that. By the way, I should just mention, if you ever want to hear, since you're going to be studying organic chemistry and maybe some biochemistry, if you watch James Tour's series on the origin of life, he just demolishes, natural origins. It's wonderful. And then our work with the prokaryote to eukaryote transition with nuclear localization signals, you see miracles there. So, but we do have data that suggests life and the fossil record are young. So if that's the case, there's not, just from a scientific evidential standpoint, you would conclude the right way to interpret Genesis is to say, well, maybe the literal interpretation is the right way because it seems to agree with a lot of facts. Now, there's still problems with the young Earth model, and I concede that, but I've seen a trend where the data has been improving over the last 40 years as we discover more. We've not gotten, I wouldn't say we're in the clear yet. It's still very difficult, and I just need, some young Earth creationists said it's undeniable. I'm like, well, no, I'm not quite there myself. I believe it, but I would feel more comfortable with more data to make an emphatic case. So, I mean, I can be emphatic about the origin of life. I can be emphatic about miracles necessary for eukaryotic evolution, for protein evolution, for nuclear localization signals. But there are, for the entire universe being 10,000 years old, there's still, we have some problems to resolve. So, so that was a long-winded question, answer to your question. Doesn't the Bible suggest it? I say, well, certainly there are many ways we could read a passage and come up with our various interpretations on the meaning. But if we're talking about historical events versus kind of a theological concept, and there's lots of that's why we have so many denominations. We have different theological concepts theologically. But if we're trying to reconstruct history, I would go with the facts and say, I think God has given us enough facts to start, at least considering special creation. And if we're undecided, I said, look, let's just wait and see, because the data keep coming in. You probably were, I kind of know how old you are, so you probably were, you probably might not have even been in high school when Mary Schweitzer discovered that there was soft dinosaur tissue, and the biological state of the materials, from things like oxidation and racemization and the presence of peptide bonds and a whole slew of other things, suggested the dinosaurs can't be tens of millions of years old, not even hundreds of thousands of years old. And I said, well, hey, look, if that's the case, then life is young, then there's no need to even try to put in an evolutionary interpretation. It wouldn't be consistent with the data. And I think this is even going to be more clear-cut than the issue about heliocentrism versus geocentrism. So is that helpful?
That makes sense, yeah.
That was a really good question. I'm really glad we're having this discussion, because you might not get this from other creationists. they would try to answer it. A lot of them will try to say, well, let's try to use a hermeneutic method or whatever. I'm like, there's a sense of that might be all moot. Are you familiar with the word moot? It's like,
No.
oh, okay, I'm glad. Okay, so it's amazing when you're a science student that we don't spend as much time on uh, uh, literary stuff. And so there are, I learned this word, so the word moot. Okay, so moot. In American law, a point where further proceedings have lost practical significance. Okay, whereas in British law, the issue remains debatable. Oh, wow. I didn't realize there was, um, that's the exact opposite. Okay, let me give you the I just learned this. I feel so stupid now. Okay, when we say it's moot, it means it doesn't matter.
Mm-hmm.
Um, okay, I'm sorry to be a little vulgar here, but a paternity test, right?
Mm-hmm.
You can have, I don't know if you've, my mom would just watch random shows, and we saw this thing called The Maury Show.
Oh, yeah, I've heard that.
Okay, so you understand. All right, so what'll happen is, you know, uh, the girl's accusing the guy of being the daddy of her baby. And then he'll give all these arguments why he's not, and she'll give arguments why he is. And it's all arguments. But what happens when the DNA test comes in and Maury says, "You are the father"? All those other arguments he made go moot. I mean, they're pointless now. Doesn't matter. It's been superseded by the genetic evidence. I'm sorry I had to pull up such a stupid. I mean, I'm sorry. I have unfortunately watched my share of trash TV. Okay, since we talked about the Maury Show, there was one time the girl was accusing the guy of being the father of the baby. When the DNA test came out, it turned out he wasn't. He did a backflip. It was so funny. Okay, you can Google backflip Maury Show. I'm sorry. Okay, so at that point it's moot. So this is going to be I know this is way more than you needed to answer your question about that interpretation, but there'll be a lot of things in your scientific discussions about creationism or anything where you can bring a fact that just renders everything moot. It supersedes all the other arguments. Supersede, are you familiar with that term? Supersedes, it takes precedence. Okay, all right. So we have about 8 more minutes before we say goodbye. You have a direct line to me via emails. But while you have me live, if you have any other questions or comments or thoughts, anything, this is your time.
Well, I did have a comment about the moot definition that you found on Wikipedia, and I'm not sure that the British thing was exactly them saying that Brits' definition of it is the opposite, but rather a jab at British people that maybe they just don't drop the subject.
I don't know. This was totally. I didn't even know there was nuances in the way I was using it. So maybe I have to find maybe I could just say this fact supersedes any other any other argument.
Yeah.
I might be more reluctant to use the word moot. So we talked to Dr. Andy, so he might know how we use the word they use the word moot in Britain.
Wow.
So I learned something today. Wow. Um, so is there anything else? Otherwise, we could wrap up the show. If you want to, hey, we have 7 more minutes, so just try to gin something up about anything.
I'm trying to think of a more sciencey question. I don't know.
Okay, how about I come up with a few of mine?
Okay.
All right. How about bad creationist arguments?
Oh, okay.
The second law of thermodynamics is a terrible creationist argument. It's more subtle than that. I'm writing a paper on that. The entropy arguments, thermodynamic entropy, bad argument. You can use entropy arguments if you use configurational entropy, but this is getting down in the weeds. So that's a really bad one. I'd say another bad creationist argument is there are no transitionals. Depends on your definition. Obviously, you can even make up your own transitionals now that you know how to use Clustal. So don't use that. So what are the better arguments for creationism?
Mm-hmm.
There is no universal common ancestor for all proteins. Are you familiar with that argument? I'm the first one to bring it up.
I guess you've kind of talked about it, but I guess I'm not really that familiar with that argument.
Okay, so let me see if I could. I'm going to give you the best. I'm going to try to give you the best ones. Let's see. And by the way, so first, I don't know if I could play this. Let me see. McCrae. Let's see if I could find this. All right, I'm going to show you a slide here, and I'm so glad you know biology because this is going to be easier because you actually do know biology. Mm-hmm. So on share. So on the left side is collagen, and I highlighted the G residues, glycines. Mm-hmm. On the right hand side is zinc finger, and especially the C's here I highlighted and the H's. You could see that these are very, very different. So if I put this through your Clustal algorithm, it's going to just puke. So it's going to say, look, there's really no significant similarity. Mm-hmm. And therefore, you can't build a phylogenetic tree between these two major protein families. That means they have separate ancestry. They have separate origins, independent origins. And I said, this is the better argument. Stop trying to, argue about, what we did with Clustal. We took the same protein across species. What you need to do is to compare one protein against the other. And then you see that they had to have separate miraculous ancestry. Mm-hmm. The miraculous part is a separate discussion. Evolutionary biologists, when I talk to them, when I show them this diagram, finally, they're like, yeah, you're right, Sal. There's no common ancestor, at least from the same gene locus. Locus meaning the same position, in the DNA. They'll still say we originated from a common cellular ancestor. And like, okay, let's just grant that for argument. But what are you going to do with the proteins? Where did they come from? Did they just sort of pop into place? And they say, I don't know. I'm like, see, that's the problem. You can't prove it, but you still believe it. But I mean, the zinc finger on the right is very complex, and so is the collagen. It may look deceptively simple, but it's very complex. Mm-hmm. That's a good creationist argument because I have quotes of evolutionary biologists having to concede that. So it's like, well, where did this come from? This is even worse to me. This is even more pointed than the nuclear localization problem. And then also, by the way, you have participated in making a very good new creationist argument. Let me get us back on screen here with the nuclear localization.
Mm-hmm.
So you actually have a good one. Are there? I would say a bad creationist argument that is moot, for lack of a better word, is how different we are from chimps. Even if we're 1% different, that's still not enough time for evolution to make those changes. The significance, like the differences in our brain capacity and our reasoning. There are some anatomical differences in our ear. So it'd be common design. And I just wouldn't focus. I think it's just a waste of time to be focusing on how dissimilar we are from chimps. I would just say the important thing is if life is young, that's all moot. It's common design. So there you go. So 3, what I consider bad creationist argument, and then we have at least 2. You're going to have. I'll give you a third good creationist argument. The eukaryotic, the emergence of eukaryotes. That's a serious problem for evolution. So there you go.
Okay.
All right. Did we have fun this whole summer? I think we did.
Yeah.
And I'll meet you backstage. I'd like to thank the audience for tuning in. I'd like to thank Katie especially for being such a great researcher and aspiring researcher. She is a researcher. She's also an aspiring researcher, and she is a student of biology. It's encouraging to see that there are bright minds out there who are applying themselves, and I wish her the very best and great prosperity and success. I'll give you the last word, Katie, to say goodbye to our audience.
Okay. Goodbye.